Okay, so it's recording now, and here's the thing. So you've got that rhodopsin, which contains the protein component and the retinal component, right? Remember that? And retinal in this case is in the conformation that is called 11 cis. Okay, when light interacts with rhodopsin, retinal goes from 11 cis into all trans. So what these terms mean, it's an organic molecule that basically changes its shape. So far, are you following me? You can just nod if you're following, or no, I don't. Okay, you're good. Okay, you're following. Now, um, what happens is the result of it is that opsin, which contains retinol in 11 cis, they become separated. So you have opsin, that's the protein component. And now you have all trans retinol. Imagine that, like, I know that it's not going to be recorded on the screen, but you can see my hand, I suppose, can you? So
Yeah.
my fist is the opsin, and this little pen is retinol. When photoreceptor visual pigment is exposed to the light, they separate. Does it make sense or not?
Wait, can you say which ones separate again?
So they are together. They get exposed to the light. Look at me. They get exposed to the light. They get separated, one from another. Does that make sense?
Mm-hmm.
Okay. So that's basically what I try to. So that's light. Okay. We good? In order to sense light again, all trans here has to be converted back into 11 Cs, and that requires ATP. So a couple of things about light reception, and then we will go to the light transduction. All right. So light reception. There are two types of cells: the cones and the rods. Right. Cones are for the bright light, colored vision, high um, resolution vision. Rods are for seeing in the dark. So far, makes sense, right? So what happens? Rods are extremely sensitive to the light. So right now, as you all are sitting in your rooms and I am sitting in mine, there's plenty of light. So virtually all rhodopsin in the rods exist in this conformation. They are separated. They cannot possibly go back because it requires so little light to get dissociated. For cones, it's different. It's kind of a balance. There's both this rhodopsin and this rhodopsin because they different, structurally different. Does that make sense to you? So you have some in your cones right now, you have some of this, which will dissociate and then will return back and will dissociate, and that cycle will provide you the ability to constantly sense the bright light. It's kind of a higher threshold of activation. Does that make sense? We good with that?
Yeah.
Yeah.
Okay, cool. Now, what I wanted to say. Yes. For the transduction. Transduction generally refers to the process of quote unquote converting a signal like light or sound into a nerve impulse. Do I make sense? Because that's what your brain interprets, not light, not sound, but the nerve impulse. So, how it works. You've got the membrane. Can you see that? What I draw? phone being away. Here you have rhodopsin. Okay. Now you're going to tell me a little bit. When light interacts with rhodopsin, what happens to the retinal?
Separate.
It converts into the old trans from 11 cis and separates from opsin, right? That separation changes the shape of rhodopsin, and that shape change activates G protein, so it's now active. That little asterisk means active. So far, you following me?
Mhm.
And that active G protein activates the enzyme that is called phosphodiesterase. So far, we're good? So phosphodiesterase is now active. Now, what happens next? Phosphodiesterase, and I'm drawing this little G protein here. Phosphodiesterase, its function is to break down cGMP into GMP. That's it. That's the function. So you have one molecule called cyclic GMP, and now you have different molecule, just GMP. Are you following me so far? Okay, we're good. We're good. All right. Let me put your face right here in the center. Okay, awesome. Now the next big thing. Remember, the levels of cGMP go down. Okay. So now we move on to talk about the channels. So we have a channel. Okay. And I'm going to go in a second. We have a channel. That's so. What was that? That is open, okay? I know why it draws in such a terrible color, but anyway, okay. Well, I guess that's it. We have a channel that is open. And what keeps it open? Do you remember? What keeps it open? Which molecule? CGMP. I can read lips. Okay, CGMP keeps it open. Now, when CGMP is gone, what's going to happen to the channel? Huh? You can unmute yourself and tell me.
Gonna close.
Yes. Amanda, are you following?
Mhm.
Okay. So now the channel is going to close. Good so far? Now, this channel normally allows ions like calcium and sodium to diffuse into the cell. When it is closed, can they go in? No. How are they charged?
Positively.
Positively. And if we have less positive ions getting into the cell, what's going to happen to the membrane potential of that cell?
Goes negative.
And if it becomes more negative, how do we call it?
Hyperpolarizing.
Hyperpolarization, yes. Amanda, so far we good?
Yeah.
Okay. Up to that moment, did you follow?
Mhm.
Brilliant. Okay. Let's move on to the next chapter in our lives. So we've got, and that is all photoreceptor cell, right? So let's see what happens when there is no light and you've got photoreceptor cell, bipolar cell, and ganglion cell. When there's no light, is photoreceptor cell hyperpolarized when there is no light?
No.
No, it's not. It's not hyperpolarized. It's normal. Does that make sense so far?
Yeah.
Since it's normal, it produces neurotransmitter. Okay, so neurotransmitter. And this neurotransmitter that is produced by photoreceptor cell, is it excitatory or inhibitory?
Inhibitor.
Inhibitory. And since it's inhibitory neurotransmitter, what does it do to bipolar cell?
Hyperpolarized.
Hyperpolarizes it. So this cell is hyperpolarized. And when cell is hyperpolarized, it is essentially inhibited. Are you with me so far? Amanda, okay, good. Since it's inhibited, it doesn't produce any neurotransmitter, and there is no signal going between bipolar and the ganglion cell. You see empty synaptic cleft. Now, light. We just mentioned what happens to photoreceptor cell in terms of the membrane potential. We just mentioned when it is exposed to the light, all those events, stimulation of G protein, breakdown of cGMP, closure of channels, what happens to photoreceptor cell?
Hyperpolarizes.
It's hyperpolarized. When it's hyperpolarized, does it produce any neurotransmitter?
No.
No, it doesn't. So bipolar cell now is not inhibited, meaning that it's depolarized. We good so far? And that depolarization leads to the release of neurotransmitter in the cleft between bipolar and ganglion cell. And that neurotransmitter that is released in that cleft, is it inhibitory or excitatory?
It's not inhibitor.
It's excitatory, exactly. It's excitatory now. And that enables the signal. Does that make sense to you? Did I answer your question?
Yeah.
Okay. I'm going to stop the recording, not because I don't want to talk to you, but because the shorter they are, the better they are produced.